Good day, welcome to the Waters and Tea Instruments Medical Device Series. This series aims to show you how we can help you understand your medical devices better from a chemical and material characterization perspective. This can then be applied to product development, to processing, to regulations, to troubleshooting, presented each week by an expert in the respective technology. For this webinar, we encourage questions, and we'll have some experts on hand to answer as many as we can in the time we have. Those we can't get to today, we will follow up with afterwards. Please post questions in the top left window and we'll answer at the end of the webinar. There are links to resources in the bottom window and a survey in the top right, which I'd appreciate you completing. I would also invite you to register for future episodes of this series. This series will comprise of multiple sessions, one a week. These will cover various aspects of medical devices and the materials used for these devices, and will highlight how some of the technology from Waters and Tea Instruments can answer some of the important questions that you have. We open the series with a focus on chemical characterization and some of the regulatory changes that have happened of late. We then moved into understanding the mechanics and strength of biomaterials and the tissues that will be replaced or surround a device, and after that, fatigue and durability testing of medical devices. Last week we discussed how to apply rheology to soft biomaterials, and this week we'll be speaking to how you can apply microcalorimetry to understanding more about your medical devices. I'm Ben McGree, responsible for the medical device markets at Waters. I'll be your host today and for each session of the series. Today's presenter will be Marlon Sakusk. Marlon is Isothermal Product Manager and Application Support for Europe, a true specialist for microcalorimetry with over 25 years of experience in product management, applications and marketing. Marlon spends a lot of her time engaged with the market, helping our existing customers, potential customers, and in general sharing her knowledge of microcalorimetry. This series covers a variety of aspects of medical devices, from the final devices themselves, to the components, to the materials used. At each stage of development, there are key challenges and activities where chemical and material characterization is crucial. Your research or testing needs might be on the device themselves, understanding the final attributes, the stability or performing quality control. Or you might be more interested in understanding the material that forms the device, how changes here impact the end performance and usage of the device. Through investigating the chemical and material nature, you'll gain a much better understanding of your medical device itself. Today we'll hear how microcalorimetry can be applied. Over to you, Marlon.
Thank you. Today I will be talking about how microcalorimetry as a technique can be used for testing of biomedical devices. The outline of the talk is a short introduction to what is microcalorimetry, and then I will give some examples of microcalorimetric application. The main focus will be on implant-related infections, but also on biocompatibility, material stability, curing, and active implantable medical devices. I will finish up with a summary and outlook. Isothermal microcalorimetry is a technique that measures heat produced or consumed during a physical process or a chemical reaction. This is done in terms of heat flow at a constant temperature with microwatt sensitivity or better. So the heat production in a sample is measured as heat flow and is directly related to the rate of the reaction. This can be seen in the left graph. So we measure the reaction or the reactions if there are several reactions. So we measure the sum of everything that goes on. If we integrate the data, we get heat as function of time, and that is the total heat produced, and it's related to the extent of the reaction. On the right, there is a scale just to show in which levels we are measuring in the microcalorimeter. So from a few tens of nanowatts up to milliwatt is the measuring range of a microcalorimeter. Why would you use microcalorimetry? It is universal. Almost all processes will generate or absorb heat. It's non-specific. We don't need to specify what we're going to look at. We can just put the sample into the calorimeter and see if there is something going on. It's simple. Sample preparation is generally not necessary, and it's not depending on the physical state of the sample. Anything that could be fitted into the sample holding ampules of the microcalorimeter can be measured on. These ampules that we see in the picture are 4 milliliter. They are 20 milliliter, even bigger and smaller ampules. So depending on the needs, you choose the calorimeter or ampule. It's non-destructive, meaning that you can do further analysis of the sample afterwards. It's direct and continuous, so it gives you real-time data. It is highly sensitive and have a high throughput. Up to 48 simultaneous measurements can be done independently of each other. It's simple to do calorimetry. So the principle of a measurement is that you put your sample in a calorimetric ampule. We see here a blue sample in an ampule. You lower the ampule into the calorimeter at the predefined temperature, and you collect heat flow data as long as required, hours, days, or even weeks. When you're finished, you remove the sample and can do other analysis of the sample if needed. This gives an overview of different applications that could be done or testing that could be done on biomedical devices. It could be effect of coating. We can study hydration and curing, efficiency of stabilizers, battery testing, like looking at pacemaker batteries, microorganism detection, which we will talk more about, general stability of materials, interactions, more also referred to isothermal titration calorimetry, material compatibility, where you may have the stability of the individual materials, but maybe they're affecting each other so they don't stay stable when they are put together. And biocompatibility, how does a material react with living cells in the body? Okay, so that was the short introduction, and now we start with the implant-related infections. 60 to 70% of all hospital-acquired infections are associated with the implantation of a biomedical device. The microorganisms that infect the body can be introduced directly during the operation, post-operatively via spread through the wound, or at later stages via the blood or the lymph systems. The bacteria that has the ability to form biofilms on inert surfaces are the most common to cause infections. These bacteria will grow and evade the defense mechanism of the host, as well as the bactericidal activities of antibiotics. Effective treatment often involves surgery, where the removal of the infection biomaterial is done and followed by a long course of antibiotic therapy. The diagnosis of implant-associated infection is difficult due to biofilm formation, and monitoring of microbial activity of surfaces or of solid substrates or inside porous structures is very difficult and often require destructive assays, where the standard methods involve culturing, staining, and they do provide endpoint measurements. Also, it's desirable to find coatings or materials that could prevent bacterial adhesion and subsequent biofilm formation. And it is important to be able to assess these antimicrobial properties of these materials and coatings, and they are difficult and rely mostly on microscopy and conventional culture. In this part of the presentation, I will now talk about how microcalorimetry can aid in this. Detection of microorganisms is achieved by their replication. Microorganisms replicate very fast. A typical bacteria will produce 2 picowatt, and with the calorimetric detection limit of about 200 nanowatt, only 100,000 of growing bacteria are required for detection. We can see an example of a dilution curve of a bacterial suspension in the lower right, where the first black curve is about 1 million of CFUs, colony-forming units, where have been diluted 10 times for each curve that comes. And in the end, we see the orange curve where there are 3 CFUs. So there are very few initial bacteria in this suspension, but they just need time to replicate and you will be able to detect them in the microcalorimeter. There is one line with 0.3 CFU, there is nothing shown, but in principle there should be, with that dilution, it should only be a bacteria in one of the three dilution that is made for that one. Isothermal calorimetric data can be treated exactly as microbiological data, where heat flow and activity will represent metabolic rates. So in the graph here, we have rates in the left column and quantities in the right column. The upper part is heat flow. You see the heat flow rate. And that is proportional to the microbial activity measured by using another type of assay that we see on the graph below. They have quite similar shape, and if we relate them to them, they will be a straight line, as you see in the lower graph. Activity assay could, example, be oxygen consumption or something to measure the activity of the microorganism. If we look at the right graph, there are quantities, and we have integrated the heat flow graph. We get the total heat. We see the extent of the reaction. It will reflect the biomass growth, and that we see in the lower graph or the middle graph to the right, where it could be biomass produced through plate counts, optical density or something that you see the number. And you can relate heat and reactants or products in the lower graph. So you see these measurements are related to each other. There are different cultivation techniques for microcalorimetry. To the left, we see just a medium that has been inoculated with bacteria that grow in suspension. In the middle, there are solid substrates or a solid medium. There it either could grow on the surface of the medium or inside the medium. And to the right, we have two examples where biofilms grow on the material that is put onto a solid substrate medium in two different ways. Either it's sandwiched onto between the material you're testing to the medium, or above a filter where it could be tested to the air. The most common complication of a joint replacement is aseptic failure, followed by prosthetic joint infection. The treatment strategies are fundamentally different, so it is important to have a correct diagnosis. Normally, tissue samples are retrieved during surgery and they are cultured, but no sampling from the surface of the prosthesis is made, and thereby the microorganisms that grow in biofilms are not detected. Diagnosis can be improved by sonicating retrieved implants to remove the attached microorganisms, followed by culture and calorimetry of the sonicates. So, calorimetry can be used to detection of microbiological growth in orthopedic implant-related infections, either in the retrieved body fluid, like synovial fluid that will be grown in a medium, as we see in the ampule to the right, or from explanted devices. If the implant is small enough, it could be put directly into the microcalorimetry and measured on, but if it's bigger, a technique like sonication culture or sonication fluid can be used, where the implant is sonicated to detach the microorganism, and then the sonicate can be used for either culture or microcalorimetric measurements. This example shows calorimetry of synovial fluid, and this is the standard synovial fluid culture is the standard method for preoperative diagnosis of a joint infection. But culture have limited sensitivity and require several days until the results. The diagnostic performance test has also been done with microcalorimetry, and it's comparable to culture, but it delivers results considerably faster. The median was 9 hours versus several days in this study. It could also be seen that all these microorganisms, they grow with different profiles. So there is also a potential to be able to identify which type of bacteria is causing the infection. There is also possibility to test antibiotic susceptibility by adding different antibiotics to the samples in the microcalorimeter to see if that has an effect on the bacterial growth. Then this is an example of sonication fluid calorimetry, where they tested different microbiological tests and compare them to the clinical diagnosis. So almost 400 implants was retrieved over a year, where they did tissue culture, sonication fluid culture, and sonication fluid calorimetry. From the clinical diagnosis, calorimetry found 96% of those infected cases, 96% of those that were clinically diagnosed with an infection. Sonication fluid and tissue culture have lower rates of detection of these infected cases. Those that were diagnosed as aseptic cases still had some infection in them on a very few percent, and these were the same for all the different techniques. So conclusion was that calorimetry is more sensitive to diagnosing implant-associated infection than is tissue culture and sonication fluid cultures. Not only joint implant has been tested. This is a case study of explanted breast implants, and this was removed implants because of severe infection, and calorimetry detected infection in less than 2 hours compared to culture that took a day. Now note that calorimetry has not identified the bacteria as such, but it has detected that there is an infection in or on these implants. The right implant reached the peak first, and this can be indicative of a higher bacterial load, as a higher inoculum will lead to faster response time. This was also compatible with the clinical observation. So calorimetry can have a benefit when it's very critical to have a fast diagnosis or test of an infection. To avoid infections, there are different strategies to use different materials or to coat in different ways to prevent adhesion or kill the bacteria or anti-biofilm formation. Most of these can be tested in microcalorimetry, but I will just give one example during this talk. Biomaterials with antimicrobial properties are now commonly used in different clinical specialties. And it's very important to be able to assess the antimicrobial effect on these coatings that is applied to the implants. This is one example where they have used the hydroxyapatite, that is a common coating to implants. So have a very good biocompatibility and function over the years, but it still have a high rate of infection associated with it. So tests were made by dope it with some antibacterial molecules, and in this case it was silver. So we can see in the graph here, the first solid line, grayish line, is the hydroxyapatite alone inoculated with bacteria, and then the 2 coming one has also been doped with silver. So we can see with silver we actually kill some of the bacteria, so it takes longer time for the bacteria to grow to be able to form these biofilms. There was also completely uninoculated, and we see that as a straight line in the bottom because there is no detection of bacteria. There is no growth of bacteria in that. The different surface treatments have its challenges. It may not be sufficiently stable. If it had drug-loaded coatings, it needs to make sure that the drug is released at controlled rate. There could be compatibility issue, and also the biofilm eradication still remains the most challenging part. I will now only focus on biocompatibility, and many coatings will, or modified surfaces will provide a superior antibacterial action, but this can however have detrimental effects on tissue biocompatibility, impairing the integration of the implants onto the surrounding tissue. And this will now lead us into the next section of this presentation, and that is biocompatibility. Biocompatibility can be measured by IMC by the principle that mammalian cells do produce heat as well. Everything that lives will produce heat. And the metabolic heat of a cell is about 30 picowatt per cell, which means in microcalorimetry we have a possibility to measure the metabolic range of between 10 to 50,000 cells. Microcalorimetry can then continuously measure this rate and see how it changes upon exposure to a material. So we can use microcalorimetry to measure the cellular response to synthetic biomaterials in vitro. There are some biocompatibility examples, and they include synthetic graft, where studies of activation of platelets or leukocytes with different synthetic materials has been done. There are dialysis membranes and orthopedic implant materials. The first example is blood compatibility with dialysis membrane. It has been suggested to work as a screening technique where granulocytes has been exposed to different membrane materials. Granulocytes are characterized by having a very low metabolic activity when they are exposed to inert materials. And when they are exposed to a material that will induce phagocytosis, they will have a very high energetic burst associated with that. So this test was done by lining the calorimetric ampules with different coatings and then put the granulocyte into that and measure the metabolic heat, the base metabolic heat. Inert material will have less metabolic heat than other material that will activate the cells. So compatibility could be ranked depending on the basal metabolism that you see after 2 hours of exposure. Another part of the test was adding chymosan particles that would trigger phagocytosis in the granulocyte. And so it was added and the measurement continued. And for those cells that had an inert material to it, they had a high energetic burst due to the phagocytosis. But those material that was already activated, had already activated the granulocytes, they have a much less energetic burst because they were already exhausted. So compatibility could be ranked either for the base metabolism or the actual metabolic burst that happens if you add the substance that would trigger the phagocytosis. This work was done some almost 30 years ago, where there was a lot of this work done, and there was a very enthusiastic approach to this. But at that time, calorimetry was not multi-channel. Well, they have 4 channels, but it was not a screening and throughput method. Today, we have 48 channels where we can do parallel testing and screening. So this method could actually be used more efficiently today to screen for these kind of things. Another example is, well, implant loosening because of bone absorption around the implant is the most common cause of failure of joint replacements today. And one mechanism of this bone absorption appears to be an inflammatory response to small particle debris that is shed from the joint implant surface and then migrate to and infiltrate the surrounding tissue. Macrophages are the main cellular type involved in this inflammatory process. It is important to get wear resistance of implants, but it's difficult, but also to control the biological reactivity of this particle. They will play a key role in the long-term implant survival. So there is a great interest in understanding the response of macrophages to the polymeric, metallic, and ceramic wear particles that are produced by joint implants. So microcalorimetry has the potential to serve as an in vitro screening tool for studying the response of macrophages to various particle variables. It may be size, type, or morphology. It can continuously measure the overall metabolic response and is suitable to detect unknown cell metabolic phenomena because we don't need to define what parameter we are going to study. We see if something is happening. There was one study made on small particles of 2 different orthopedic implant materials, ADPHE and cobalt chrome alloy, that were exposed to macrophage cell suspension. Also, LPS was used as a positive control, which is a bacterial endotoxin that will induce a response in the macrophage cells. It was seen that there was a significant heat flow rate differences for the macrophage cultures with endotoxin or these particles compared to cell cultures without stimuli. The response was stronger for the polymeric particle compared to the alloy particle, and the conclusion of a few of these papers was that microcalorimetry can be used to detect the heat associated with the phagocytosis of particular materials. So that's one part of the presentation, and now we will go from the living metabolism to the actual material. So now we come into the material stability, the physical-chemical stability of the material. Assessment of the stability of the synthetic biomaterial is of vital importance for the prediction of the lifetime, both during self-storage and in vivo. The synthetic material can be subjected to oxidation, corrosion, or hydrolysis under conditions that occur in the body. These reactions are slow, and they will also slowly change the mechanical properties of the implants. So it's important to know this. Heat flow calorimetry, or heat flow, is related to reaction rate. We have heard that already, and it's been used for stability assessment of a lot of different materials already, and it can also be used for implant type of materials. And it's well suited to rapidly be able to quantify the rate of slow changes in materials because of its high sensitivity. It can directly observe and quantitate the rates of degradation of as low as 1% per year in a solid material at body temperature. There are different types of stability assessment that you can do. Storage stability, stability in simulated body implantation conditions. We can look at effects of sterilizations, effects of coatings, and we can look at compatibility, which is then if you combine different materials and they affect each other. Ultra-high molecular weight polyethylene is widely used as one of the bearing surfaces in joint replacements. It's known that the pre-implantation sterilization process has a strong influence on slow but steady oxidative processes, and this will lead to embrittlement and compromising the mechanical integrity and thus the clinical performance. Specifically, gamma irradiated material creates free radicals in the material, and addition of antioxidants as alpha tocopherol is done to quench this oxidation process. We see here stability testing as function of storage stability. In the first one, we have different sterilization methods. The control that's unsterilized and ethylene oxide or gas plasma sterilization yield almost no reaction at all, but samples that has been gamma irradiated both in nitrogen and air show much higher reaction rate in the microcalorimetry. Now you can see that this is measured for a very long time. Typically you do not need to measure for this long period of time, but to learn about the material, sometimes they are measured for long periods of time. And then it's the right example is simulated implantation condition where it's at 37 and with a buffer of a pH, body pH. We can see that the reaction rate has gone up slightly also for the control and the GP and ETO sterilized material, and the reaction rate in the gamma radiation is still higher than the other. So how can we explain the differences here with the different gamma irradiation? Well, we can do the test under different atmosphere, and simulated shelf storage results will imply that with gamma sterilization we get roughly 10 times as many oxidizable bond compared to case when you use the ETO or GP sterilization. But sterilizing in nitrogen does not prevent the creation of the oxidizable bond, but it will just push the oxidation until the material actually is exposed to oxygen. And at the implantation time, the material will be exposed to oxygen, if not prior to that. So this can be seen if that is taken into account, and they almost fall together on the same line. So benefits of using microcalorimetry for stability in lifetime prediction is speed. You can get rate constants within a few hours or maybe days for slow processes. Sensitivity, it's not very much sample that is needed. We get a direct determination of kinetics. It's non-destructive. Samples can be further analyzed afterwards. And the sample throughput. 48 simultaneous measurements is possible, so screening and different formulation and sterilization methods can be done quickly and reliable. And an example on curing. The example on curing would be on bone cement. And the reaction rate of calcium phosphate-based cement is of great importance for the clinical use. It should not harden too fast to allow molding or injection, neither should it harden too slowly. And too fast setting or curing will also lead to a lower strength and toughness of the cured material. Microcalorimetry is an excellent tool to study the effects of the setting reaction, and you can test with different additives to improve strength or other properties of the bone cement. There are examples of looking at influence of particle size or milling times on the setting reaction kinetics, effects of adding citric acid as a retardant on the setting time. The setting time effects on the mechanical properties are measured by indentation methods, and they are strongly correlated with the maximum heat flow. So calorimetry and mechanical properties are related. For acrylic-based bone cement, the setting time measurements are also possible with microcalorimetry, and these studies suggest also that the sterilization method has important effects on the cement stability. There are two ways of studying curing in the microcalorimeter. In situ, where you measure the instant reaction when cement is mixed with water. That you can do inside the calorimeter, and this is important for some bone cement types that will set within minutes. And then you can follow the setting behavior and strength development over time. Some bone cements will take hours or even days to reach its maximum compressive strength, and we can follow the whole process continuously in the calorimeter. For some load-bearing situations, it's also important to rapidly reach high mechanical properties, and studies of composition to better be able to control the strength evolution is of great interest. So the benefit of microcalorimetry is that the curing process can be followed continuously from start to end over extended periods of time. The example here shows two examples or two types of calcium phosphate cement, and you can see they cure completely different. One is very fast and one is slower, and it's also cure over a longer time. And the final example is on active implantable medical devices, which could be pacemaker batteries, and it's very important that these work without any problem once they are put into a patient's body. So tests can be made for small internal short circuits. It could be done on the battery alone or a battery in a well-defined circuit. Typically, this measurement is done as steady state heat flow of a pre-equilibrated battery. So it's equilibrated at 37 degrees and then put into the calorimeter, measure heat flow and remove it. On the scale here, we're measured for a long period of time, but usually it's just steady state and out of the calorimeter and test an S battery. Good batteries should have below a certain level of heat flow, and bad batteries will have, if there is a short circuit in the battery, it will give up much more heat. So, summary and outlook. We have talked about implant-related infections and how microcalorimetry can detect infection fast, and it could be used to evaluate prevention strategies. For the future, we can also see that identification and antibiotic susceptibility testing can be performed. For biocompatibility, it has a high sensitivity and it's a continuous and general method. You can also see for future that tissue engineering, we can measure on how tissue grow for tissue engineering applications as well. For material stability, it has very high sensitivity and you can get kinetic information very fast on measuring stability on materials. That has been focused that you want to have stability materials, but I can also see in the future to look at biodegradable material to optimize the degradation of a material under different circumstances. We talked about curing, that we can continuously monitor setting profiles, and an example of active implantable medical devices, which is a non-destructive technique. Thank you.
This series will be talking to many aspects of medical device and material properties. How it can help you measure, and how it can help you understand your material better. Next week's tour is the last in our current series, and will be on applying thermal analysis to medical device materials, presented by Phil Davis. We hope to see you then. We'll now take some time for questions. Whilst we collate the questions that you've been submitting during the talk, we'd appreciate it if you could answer a short poll to help us prioritise how we can help you better. Okay, so now we'll have some questions. So first question for Long, will oxygen not be limited for the microorganisms test?
Yes, that could of course be an issue if you have a closed ampule and you have a limited atmosphere of that. But for those tests that we have been showing, time to detection is faster than the limitation occurs for the oxygen supply under those conditions that the experiment has been performed. If you use bacterial growth, or you do bacterial growth testing in a liquid media, there is a very slow diffusion of liquid from the headspace into the medium itself. But usually you will be able to detect the bacteria before that is limited. If you would do a solid medium and you have the bacterial growth on the surface, then the bacteria have more oxygen supply from the atmosphere, but then there could also, of course, be limitations for nutrients from the media because the diffusion is low. But they have shown in the tests that usually we see the detection of the bacteria before this is an effect. But it's also been shown if you measure for a long time and many bacteria, you can see the switch. You can actually detect when the bacteria go from aerobic growth to anaerobic growth. You will see a shift in the profile during that. So that's also one thing that could be seen by measuring in the microcalorimeter.
Excellent, thank you. We have another question here. What is the minimum bacteria number detectable via microperimetry reproducibly measured?
There is in the literature several measurements. It depends on the bacteria species as well. But in many cases, you have put in 1 to 10 CFUs of bacteria, and you get them reproducibly. You get detected after a while, but the fewer you have from the start, the longer it will take you to detection. So if you start with 1, it will take more hours to get to a detectable limit. But there are very, very few cases I have read about, at least, that you don't detect bacteria if you have them to start with. And the profile you have seen on the examples are usually very reproducible when you get the bacteria out, depending on the starting number that you have when you started the test. And we talked about the bacteria that will generate about 1 picowatt per bacteria. So you need to get up to maybe 100,000 before you detect them, but you can start with a lower number and still detect them in the end. I hope that answers the question.
Okay. Yes. So yes, it says thank you very much. So yes, it did. Thank you. So we've got a few questions about the slides. So what we'll do after this webinar is you'll get a link and a follow-up email with a link to the recording. For those of you that want the PDF, we can arrange that as well. So we have another question here. Is there any method or standard developed using MC for determination of antimicrobial material efficacy?
Today, I don't think there is any standard method how to do it. It's still a lot in research, but there are a lot of publications of research groups that are developing these techniques. So there are some procedures that you try, but they are still under development. There is no official standard for how to do this test yet. They are used in clinical applications in some places, but they are used in parallel as now together with more traditional techniques. But we hope this will be more of a standard in the time to come.
I have a question on, can one sterilize the equipment?
Yes, we have the sample handling system, so it's completely contained with your sample. Those are usually disposable glass ampules with crimp cap ampules, and those can be sterilized before the measurement. We also have other type of equipment that's non-disposable, but everything that contains a bacterial culture or something can be sterilized upon use.
Okay, I've got another question here. Is there a way to change conditions during the test?
Yes. Again, as I mentioned, most of them are closed ampule measurements, but we do have other types of sample handling systems as well, where we can change the conditions during the test, which could be of advantage if you would like to flush a certain atmosphere over the sample during measurement. That is possible, both for looking at providing fresh media or fresh oxygen over a growing sample, or if you want to change atmosphere when you are doing oxidation tests, so you can switch between oxidation or nitrogen, or air or nitrogen when you do stability tests, for example. So we call them microreaction system, and you can change oxygen, you can make injections, and you can also stir your systems during measurements. I've not shown anything in this presentation, but the possibilities are there. So that's the answer to that one.
Thank you. The question here, which is, how can bacterial identification possibly be achieved?
Yeah, you saw on the pictures that we have many bacteria will have a specific growth profile, but it's also possible to add a certain substrate or a certain media that some bacteria will grow on and some bacteria will not grow in, or add an antibiotic that hits some types of bacteria but others don't. So it's possible to change the environment for the bacteria to be able to detect that specific bacteria you would like to do, or screen for bacteria by having ampules, like we have the 48 channel, that you can have different media in different ampules and compare how they grow in different type of media or substrate. There is also ongoing work on trying to model the data to be able to see the growth profile and how it looks like, what it could be for bacteria by treating the data in different ways, as well as if you have mixed cultures, also to deconvolute, which if you have several different strains of bacteria growing in the same mixture. So there is work ongoing on that, and that's quite exciting work to be able to see that on growth profiles.
Excellent. So an area to follow up on maybe when that work's been developed. So I think we'll call it a day there. So thank you to everyone for attending. Thank you very much to Tamara for presenting. It was a very interesting talk today. We have one more in this current series next week. Please do join us then. And as I said, we will send a follow-up email to this talk to send a link to the recording. And for those of you that ask questions, if we haven't got to, we can follow up directly after this event. So thank you very much, and goodbye.
Thank you.